Today we're so interested in this, the study of how some of these extreme climate events that we've been seeing, how they affect human societies. Do they affect them, how do they affect them over a long term? How do they affect them over a wide geographic region? How does the same climate event play out in lots of different parts of the planet? Well, today, of course, we can watch that evolve when an event hits, but what happened in the past? Tree rings provide this incredible way to do that because they're these kind of passive sentinels recording time and lots of other things to go with that time. So in each time capsule that's locked away in a tree ring sequence, we have all kinds of information. We have the radiocarbon that was in the environment at the time that tree ring formed. We have maybe oxygen isotopes, maybe other carbon isotopes that tell us about rainfall, that might tell us about temperature. We can know that a tree is putting on a ring every year. And the amazing thing is that if we start with today, if you or I, if we went outside and we took a sample from under the bark of any of the trees that we see outside, we would find a ring forming, and it would be this year's ring. So we know the date, and so we know the date of the one before, and the one before, and the one before, and the one before. And the neat thing is about tree ring research is we can overlap these patterns back 12,000 years dated from today, all the way back, each year represented by a single ring. And so that's an amazing way to measure time, and it's an amazing archive. And all the time we're finding new ways to unlock the secrets of the tree rings.
This rule, the hydra, not the mythical beast with all the heads. This hydra is a freshwater polyp that's a centimeter long and reproduces both asexually and sexually, can regenerate after it's been cut into pieces, including being able to regenerate its entire head, and as it turns out, is pretty much immortal. Not bad, being immortal and stuff, but I would like to see it try to host a YouTube show. The hydra has fascinated scientists for centuries, and when they sequenced its genome back in 2010, they discovered that it had the same number of genes as humans. We also have a surprising number of genes in common, including certain genes associated with Alzheimer's and Huntington's disease, so we've been studying these tentacular little guys ever since. Because, maybe they can magically repair the neurodegeneration that leads to Alzheimer's. That would pretty much be exactly what we would expect from hydras at this point. Why not? Because we've been watching hydras for so long, scientists have suspected for a long time that they don't senesce, but this was the first large-scale study that focused on finding out for sure. The team followed more than 2,000 individual hydras for a combination of 3.9 million days. The hydras they studied were between 0 and 41 years old, and they observed them over a period of 8 years. The result? No decline in fertility or increase in mortality was observed at any point in the hydra life cycle. Although, if they don't age, it's not really a life cycle, is it? It's more of a lifeline that gets cut off when they get eaten by something. And that is what I have been able to do that most hydras can't. Avoid getting eaten, so far at least. These hydras reproduced, or budded, about once a month, and they died at a rate of about 1 in 167 per year, no matter how old or young they were. If the mortality rates observed in this study continued, that means that in 3,000 years, 5% of those hydras would still be alive if they were kept safe in the lab. That would be a really long experiment. So, how do hydras not decline with age? Well, there are a lot of factors that might contribute. They have a relatively simple body plan and relatively few different types of cells, which probably helps. But the most important factor is most likely that hydras constantly produce new stem cells. Every cell in a hydra's body is replaced every 3 to 4 weeks by fresh cells produced by brand new stem cells. It's essentially regenerating once a month, and in a completely different way from the jellyfish we talked about earlier, which reverts to an earlier stage of its life cycle when it's stressed. There's a lot we might be able to learn from this. What we now know about hydras suggests that they could hold the keys to big breakthroughs in research on stem cells, aging, and diseases like Alzheimer's. And while we're at it, maybe we should leave little notes in hydra's genetic code for any future aliens who might one day find our planet. Maybe they'd sequence the genome of our immortal little buddies and it would say, "We were here, signed humanity." And then we could leave a link that they could follow to the last working internet server, which would just be Rick Astley's Never Gonna Give You Up. Thanks for watching this episode of SciShow dinosaur news, and thanks especially to our patrons on Patreon like Emily Burrows and Mike Boyle, who make this show possible. We just picked a couple to say thanks to them specifically. If you want to help us keep making videos like this, you can go to patreon.com/scishow, and don't forget to go to youtube.com/scishow and subscribe.
Hans Spemann and Hilde Mangold were pioneers of developmental biology. They showed how the pattern of the embryo is created by interactions between one group of cells and another. In 1924, they made a famous discovery. They found that a small piece of tissue called the organizer, taken from a specific site in the early frog embryo and transplanted to another embryo, could control the behavior of neighboring cells and direct the formation of an entire body axis. The key experiment is reenacted here by a modern developmental biologist using the frog Xenopus laevis. Two Xenopus embryos are maneuvered under the dissecting microscope. The embryos are beginning to gastrulate. The blastopore, where cells are tucking into the interior, is visible as a dark crescent in the embryo on the left. The dorsal lip of the blastopore contains the organizer cells. With a pair of forceps and a fine tungsten needle, a block of organizer tissue is cut from the embryo on the left. Using a hair plucked from a human eyebrow, the block of tissue is gently pushed into a site on the ventral side of the other embryo. An hour later, the graft has healed into the host embryo, and the organizer cells have been integrated at an ectopic site. 2 days later, the host embryo has developed into conjoined twins. The grafted organizer has caused the host cells around the graft to form a second body axis, complete with central nervous system, eyes, somites, and other structures.
The oldest animals in the world are the giant tortoises on Galapagos, aren't they? Admittedly, they can live up to 200 years or more, but the oldest living animal on the planet is found in the seas around Iceland, and is indeed named after Iceland, Arctica Islandia. It's the humble black clam, or Icelandic cyprine or quahog. These three old codgers are over 300 years old. The clam's way of life is quite unusual. It digs down into the seabed, particularly in the winter months, where it can stay hidden for months on end, up to 20 centimeters below the seabed. It seems to be able to stop breathing and exist in a state of suspended animation for days on end. There are other creatures that like growing about in the sand as well. Here we see a little amphipod digging its way. Here we see clams mating. The male ejects his sperm and the female her eggs. This far from intimate procedure seems to go on with no contact at all, at a distance of 5 to 10 meters, and it's difficult to see how they even sense each other's presence, let alone be sure that the eggs can fertilize. Clams mature between the ages of 10 and 20, but occasionally from the age of 6. Normally their nuptials are confined to the summer months, but these two have chosen to get hitched on the last day in January. The oldest clam ever found, and therefore the oldest animal that has ever lived, was discovered near the island of Grimsey a few years ago, and was estimated to be around 507 years old by the Bangor School of Ocean Sciences. It was christened Ming, as it was born around the time of the Ming dynasty. It predates the Black Death in Iceland. It's quite difficult to discover any particular pattern of behaviour of clams. Scientists have monitored the behaviour of clams, for instance in Eyjafjörður, and it has come to light that clams can be found in the sediment at different depths in all seasons. Generally though, they were found nearer the surface in the summer months, which is the time when they are most active, feeding, filtering single-celled algae from the ocean and breeding. They can be found in considerable numbers, up to 7 kilos per square metre of sand. Usually they can be found at a depth of around 20 metres, though they have been found in a couple of metres depth and up to 200 metres down. The clam stocks around the coast of Iceland are estimated to be over 1 million tonnes. They were fished in some quantity in former years in Thorshab, but very few are harvested today. Your only hope of getting a taste of this delicious shellfish these days is to get in the good books of one of the divers.
Autophagy. Why do we need it? This is Paul. Every day he reads his favorite newspaper, but over time a giant stack of paper collects in his house. Only the recycling truck can save him from drowning in yesterday's news. But what happens to his paper afterwards? Much of it is reconstituted for new uses in the cycle we call recycling. But long before we began recycling our trash, nature was following the same principle. Even the cells in our body recycle their trash. There, the cycle is called autophagy. Cells eliminate invading viruses and bacteria, but also harmful protein aggregates and old or damaged parts of the cell's nucleus that are no longer needed. The process of breaking these things down involves two liquid-filled structures inside the cell. The autophagosome collects the trash and transports it to the lysosome. The lysosome contains digestive enzymes that finally separate the cellular junk into its component parts. But don't worry, nothing goes to waste. These broken down parts are turned into new cell components that can be used again. This has many advantages. Our body's natural resources are conserved, we save energy, and our cells' trash can never overflow. But sometimes the trash isn't disposed of correctly. This can cause diseases such as infections with certain forms of viral influenza, Alzheimer's, or cancer. But how is autophagy controlled? Paul found the answer in one of his newspapers. The Japanese biologist Yoshinori Osumi was doing research about 25 years ago when he made a major discovery. He was investigating the genes of simple yeast cells. In various experiments, he kept changing those genes in order to test the effect this would have on the cell's recycling process. Eventually, he found 15 genes in the yeast that were responsible for different stages of the autophagy process. Then he was able to apply his finding to human cells, which is why he received the 2016 Nobel Prize in Physiology or Medicine. There are still many puzzles and secrets about autophagy, but scientists are working on decoding them. Paul is relieved to know how important his built-in recycling system is for keeping his cells healthy.
The ability of cancer cells to avoid apoptosis, or programmed cell death, and continue to proliferate is one of the hallmarks of cancer and is a major focus of cancer therapy development. Two signaling mechanisms exist for activation of apoptosis: an intrinsic mechanism and an extrinsic mechanism. In the intrinsic pathway, cell death may be triggered through DNA damage or severe cell stress. Following activation, intracellular pro-apoptotic proteins are released to activate caspases. This ultimately results in apoptosis, a process commonly blocked in tumour cells. Restoring the intrinsic apoptotic pathway after it has been inactivated may promote tumour cell death. The extrinsic pathway is activated in response to diverse external signals, such as APO2L trail. This pathway has the potential to induce death of tumour cells independently of the intrinsic pathway. Development of novel molecules that promote apoptosis by targeting both pathways of apoptosis, intrinsic and extrinsic, advances the understanding of the mechanisms behind tumour cell proliferation. Genentech Bio-Oncology is committed to developing novel molecules that promote apoptosis by targeting the intrinsic and extrinsic pathways.
In healthy people, all sensations, movements, thoughts, memories, and feelings are the result of signals that pass through billions of nerve cells or neurons in the brain. Neurons constantly communicate with each other through electrical charges that travel down axons, causing the release of chemicals across tiny gaps to neighboring neurons. Other cells in the brain, such as astrocytes and microglia, clear away debris and help keep neurons healthy. In a person with Alzheimer's disease, the most basic form of dementia, toxic changes in the brain destroy this healthy balance. These changes may occur years, even decades, before the first signs of dementia. Researchers believe that this process involves two proteins, called beta amyloid and tau, which somehow become toxic to the brain. It appears that abnormal tau accumulates, eventually forming tangles inside neurons. And beta amyloid clumps into plaques, which slowly build up between neurons. As the level of amyloid reaches a tipping point, there is a rapid spread of tau throughout the brain. But tau and beta amyloid may not be the only factors involved in Alzheimer's. Other changes that affect the brain may also play a role over time. The vascular system may fail to deliver sufficient blood and nutrients to the brain. The brain may lack the glucose needed to power its activity. Chronic inflammation sets in as microglial cells fail to clear away debris, and astrocytes react to distressed microglia. Eventually, neurons lose their ability to communicate. As neurons die, the brain shrinks, beginning in the hippocampus, a part of the brain important to learning and memory. People may begin to experience memory loss, impaired decision-making, and language problems. As more neurons die throughout the brain, a person with Alzheimer's gradually loses the ability to think, remember, make decisions, and function independently. Achieving a deeper understanding of the molecular and cellular mechanisms and how they may interact is vital to the development of effective therapies. Much progress has been made in identifying various underlying factors. Advances in brain imaging allow us to see the course of plaques and tangles in the living brain. Blood and fluid biomarkers are providing insights about when the disease starts and how it progresses. More is also known about the genetic underpinnings of the disease and how they can affect particular biological pathways. These advances enable the development and testing of promising new therapies, including drugs that reduce or clear the increase of tau and amyloid proteins in the brain, therapies targeting the vascular system, glucose metabolism, and inflammation, and lifestyle interventions like exercise or diet, and behavioral approaches like social engagement that may enhance brain health. Research is moving quickly, ever closer to the day when we can delay or even prevent the devastation of dementia.
In many cultures, surnames are passed from father to child. I am a MacLysett because my father, my father's father, and my father's grandfather were MacLysetts. Unfortunately, my many female ancestors don't get recorded, and in fact, I don't even know my great-grandmother's maiden names.
But there does exist a very important genetic link between the women in my family, and that's found not on the 23 pairs of chromosomes that lie within the cell nucleus. Instead, it's found in another part of the cell called the mitochondria. So here it is, this is a mitochondria, and this is a component of cells of eukaryotes. That's everything except bacteria. They're sometimes known as the powerhouse of the cell, and that's because they produce all the chemical energy, known as ATP, that's required for metabolism. But they do other things as well. They're involved in signaling and in cell growth and cell death. But one really fascinating thing about mitochondria is that they've got their own little circular genome that's completely separate from the other chromosomes in the cell. It's really very short. It's about 15,000 times shorter than chromosome 1, and it only contains 37 genes, 13 of which are involved in the energy functions. And in any one given cell, there are many mitochondria, ranging from hundreds to hundreds of thousands. But the really fascinating thing is how they got there in the first place.
In 1966, a young biologist called Lynn Margulis noticed similarities between mitochondria and free-living microbes and proposed a theory on the origins of the mitochondria. She suggested that about 1.5 billion years ago, one bacteria was eaten by another cell and instead of being digested, lived inside the other cell and formed a mutually beneficial symbiotic relationship, eventually becoming the mitochondria.
When Lynn Margulis first proposed this theory, she wasn't believed, and the idea failed to gain traction. She showed remarkable tenacity, however, and she stuck with it. Eventually, her perseverance paid off, and it was later shown that the mitochondrial DNA has similarities to the DNA of free-living bacteria. Her endosymbiotic theory was proven true. Disappointing to think that the reason why this idea was slow to gain acceptance is the fact that it was championed by a woman. These days, however, it's no longer considered a crazy idea, and it's in every biology textbook. But the mitochondrial DNA has also helped women be remembered in another way, and that's because of its special inheritance pattern. And this is that the mitochondria are provided by the egg cell, but not the sperm. And so much like surnames are passed from father to child, mitochondria are passed from mother to child. And more than this, the mitochondrial DNA doesn't undergo the same recombination as occurs to the other 23 chromosomes. So your mitochondrial DNA was inherited intact from your mother, who got it from her mother, and so on. And so this provides a record of the maternal line right back to the origins of humans. So to me, there's a certain poetry in the way that the mitochondria links us to our female ancestors, while at the same time serves as an important reminder of the challenges faced by women in science.
or something like it, to survive dried-up ponds. The African turquoise killifish jolts to life following a phase called diapause. It's a state of suspended development that helps organisms survive extreme environments. Diapause exists among other vertebrates, such as roe deer, bears, and even lab mice. And it has led scientists to wonder, do organisms age at all during the process? New research into African killifish has shown that its period of diapause during embryo development has no negative effects on the lifespan of a fully developed adult. Turns out, the African killifish is a perfect candidate to help answer the diapause lifespan conundrum. Its short lifespan of 4 to 6 months coincides with the wet seasons in countries like Mozambique and Zimbabwe. So the African killifish species has evolved to have its embryos enter diapause so that they survive the extreme dryness that follows these rainy periods. Unlike in mammals, killifish embryos grow outside of the animal's body, which makes them easier to experiment on. For their experiment, scientists established 2 groups, one that hatched without entering diapause, and one that hatched after experiencing 5 months diapause. Surprisingly, after hatching, both groups lived roughly the same length of time, which led the researchers to conclude that diapause had little to no deleterious effects. Past research discovered that diapause delayed aging with invertebrates, such as insects and the worm C. elegans. But this African killifish research is unique, since the killifish is a vertebrate. After observing how diapause occurs naturally in killifish, the researchers went the extra mile. They investigated how the perturbation of specific genes might affect the embryos during diapause. They found that knocking out a gene called CBX7 caused embryos to fail to maintain diapause, highlighting the importance of specific genes in maintaining this suspended state. The researchers believe these findings could help discover general mechanisms that preserve cells, such as stem cells, for long periods of time. This could also inform strategies to combat aging and age-related diseases. In other words, studying how the African killifish pauses its clock during development could perhaps identify ways to adjust the aging clock in other species, including humans.
For as long as humans have been around, people have been imagining and dreaming about ways to live longer. You know, the proverbial fountain of youth. In our study, we found that the unaffected kindred had a median lifespan of 75 years. And the carriers of the mutation had a median lifespan of 85 years. We were able to interact with and investigate and study nearly 180 members of this Amish community, 43 of which were carriers of this very, very unique mutation. Telomeres tend to get shorter as we age, in all of us, and it predicts how long you're going to live. It predicts if you're going to get cardiovascular disease. They predict if you're going to get cancer. They predict if you're going to develop diabetes down the road. We found that the carriers of the mutation, they look like they're completely protected from the development of diabetes. The Amish tell us a lot about the potential safety of a drug. So that gives us some reassurance that it's going to be safe to give people for a long time.
Cloning 101. The ability to create a clone used to be science fiction. Dr. Ian Wilmot's group changed that in 1997 with the creation of Dolly the sheep. Since then, the debate on applying the technique to clone human beings has been ongoing. Based on all the discussion, it may sound like cloning, human or otherwise, is an easy process. Find out what is actually involved in the cloning process by viewing our short animation. What are clones? Simply put, clones are organisms that have identical genetic material. In other words, the sequence of bases in their DNA is exactly the same. Long before the birth of Dolly the sheep, clones had been observed in both nature and in the laboratory. When a couple has identical twins, or triplets for that matter, the children are clones of one another. A plant cutting can also be used to generate a clone. Prior to 1996, it was thought that cloning an entire animal could only be done with embryonic cells, cells present in the early stages of an organism's development. In the 1950s, scientists generated entire frogs from embryonic frog cells. After a small number of cell divisions, embryonic cells start to change into different types of cells that an organism needs, including cells that form muscle, blood, liver, etc. This process is called differentiation. Although each of these cells has the same genetic material, each cell can only access the genes needed for its particular function. Before the experiment at the Roslin Institute, it was thought that once cells differentiated, they could not be used to generate an entire organism. For instance, in a sheep, udder cells could generate other udder cells, but not an entire sheep. The scientists at the Roslin Institute solved this problem by growing sheep udder cells under starvation conditions. This put the cells in a state similar to embryonic cells. This is called the G0 state. An egg cell was taken from another sheep. The nucleus, which contains the genetic material, was removed from the egg cell using a very fine needle. They then used electric shock to fuse one starved udder cell with one nucleus-free egg cell. They made 277 of these fused cells. Although the egg cell came from a black-faced sheep, notice that the nucleus with the genetic material came from the white-faced sheep. These fused egg cells were then inserted into several different sheep. These surrogate mothers were also black-faced. Of the 277 fused cells, only one progressed to form a developed lamb. Dolly was born on July 5, 1996. Scientists found that Dolly had the same DNA as the udder cells she came from. She is a clone of these udder cells. Dolly has given birth to a lamb named Bonnie, produced the natural way. Other lambs have been born at the Roslin Institute through their cloning process. Some carry genes that will produce usable human drugs. A laboratory in Hawaii, run by Dr. Ryuzo Yanagamachi, was the second group to successfully clone an animal from an adult cell. They cloned mice using cumulus cells, a cell type found in the ovaries. The cloning method used by the lab in Hawaii was different in two ways from the method used to clone Dolly. First, the cells used to clone the mice were not grown in culture, but instead were used immediately. Second, the nucleus was removed from the cumulus cell and then directly injected into the egg cell. This egg cell's nucleus had already been removed. The Yanagamachi lab used coat color to track genetic heritage. The cumulus cell comes from an agouti, brown mouse. And the egg cell comes from a black mouse. The egg cell now had the same genetic information as the nucleus donor mouse. The egg cell was then activated and implanted into a white host mother. On October 3rd, 1997, the host mouse gave birth to Cumulina, named after the cumulus cells she was cloned from. Cumulina is the same color as the mouse that donated the nucleus. DNA fingerprinting confirmed that Cumulina had the same DNA as the nucleus donor. The scientists have taken cells from Cumulina to make more clones. They have successfully made several generations of clones, and all mice seem normal. Dolly the sheep died at the age of 6. Since the world said hello to Dolly, several other animals have also been cloned. Both Dolly and Cumulina were cloned from cells in the female reproductive system. Cows have also been cloned using ovary and cumulus cells, with the same method that was used to clone Dolly. Pigs have been added to the cloned animal menagerie. Scientists hope to use cloned pigs to grow organs that can be transplanted into humans.
Of studying something that was completely unknown. I just like that. And this was unknown, but I mean the mechanisms and whether you could even get long-lived mutants that were really not just trade-offs or something, but were really interesting long-lived mutants wasn't known at all. And so I started doing that when I was a professor at UCSF.
So, I mean, if you said that one of the things that worried you was that you were going to age too.
Yes. I didn't stop it either. Look at happened.
But what was your what was the underlying belief that what you discovered in this little tiny millimeter or a couple of millimeter long nematode worm was going to have any relevance to human aging?
Because everything that the worm did, everything seemed to be done the same in other animals. In fact, when I was there, that's when the whole field was discovering that the mechanisms for development and pattern formation were conserved. They were the same in all animals. Even though the patterns were totally different, the mechanisms to make them were conserved. So I thought, and not only that, people knew that by that time that the cell cycle mechanisms, actin, myosin, everything. So there was this new idea, really, that there was nothing that would be totally a worm thing. So I figured if you could find something out in the worm, there would be a good chance.
Okay, so you've got a genetic system, and you think it's going to be similar to humans or mammals. Is there some sort of principle of what you've discovered that's sort of reasonably easy to describe about what underlies aging and how one might be able to manipulate that for longer life?
Yes, I think the take-home message, which I tried to stress in my talk too, is that animals have the ability to live longer than they do. They can protect their cells and keep their proteostasis levels at high enhancement, and but they don't usually bother to do that any more than they have to. And so what happens is that under conditions of stress, they roll out these protective mechanisms so they don't die. But then that's it. And what we've done is to just we've switched them on even when the worm would not have us switch them on. When conditions are good, we switch them on. And so now they become resistant to everything, even though they have their normal life, and they live, much, much longer than they would normally live.
So you now work at Calico Life Sciences, which is a Google company set up to think about longevity or to tackle aging. Are you an optimist? Do you think there is a possibility that what you've learned in worms, I mean, you've told us that the biology is very similar, but that's a long way from saying that you might be able to manipulate human aging in a similar way.
Well, I think that finding that, three things, that the same genes affect aging in flies and mice are vertebrates like we are. And flies says it's very ancient and conserved, and so we should have it. The mechanism. That's the first thing. Second thing is that there are these hints that this network affects aging in people. So for example, if you probe a GWAS study with not just one gene but the network, you get a nice signal, for example. And there's a lot of other little. So I think, yes, I think that. Personally, I think the chance is really, really high. Whether you could do it, though, with no side effects, which is critical, I don't know that. That's really tricky. I'd like to know more about small dogs. Small dogs weren't bred to live long. They were bred to go in little holes or do whatever, sit on laps or whatever. But they are long-lived. So it would be, and we know they're IGF-1 mutants from sequencing, and they have low IGF-1 levels, but we don't know whether that's all. You know, if you just took a dog and you just only made that change, what would happen? Would it be a nice little, you know, terrier or a chihuahua, or is it all, or are there other things that you need to, you know, to suppress side effects? I don't know.
So the small dogs are actually using the same pathways are involved in making the small dogs live a long time as the worms that you discovered earlier.
Yes. In other words, they live long and they are IGF-1 mutants. And in worms, if you are an IGF-1 mutant, then you live long. So in worms, we have the causal relationship. And in dogs, it's possible that for some reason in dogs that's not the cause, or some other cause. But it's the simplest explanation.
In your lecture, you told us that there are some worms that can live 10 times their normal lifespan. Yes. So are we going to see a human who can live to 750 or 800 years?
Who knows?
Do you think that is there? I mean, or more seriously, do you think there's a sort of upper limit on on the human lifespan? Is there any way of thinking about what that might be?
Well, I don't know. I mean, we live already a long time, and I can imagine one thing that we've maxed out. That's it.
Yep.
Or I could imagine that we live as long as we do, and not longer, so that we can take care of the grandchildren. Because most people die when the grandchildren go into college, more or less. That's about the time your grandparents are dying. It just is, usually. So, and that's long enough for them to help the parents out to raise the grandchildren, and they do that. So maybe that's why we live as long as we do, maybe. And why we don't live longer. I mean, who needs you to take care of the great-grandchildren if you have grandparents around? You don't really need them. So maybe that's why. And if that is the reason, which it may or may not be, and I don't see, I mean, of course we'll never be immortal because you'll be hit by a truck. No matter if there's any probability, by time's enough time, that's it. You know, so that will never happen. But yeah, I don't know. The other thing is that I think it was so stunning to find mutants that you know, with one gene change that could stay young in all respects so long. I mean, I never thought that would be possible, a doubling of the lifespan. Intuitively, it doesn't seem possible for humans, especially since there are no 200-year-old people that we know of. Especially, that could be a real, maybe there's a reason for that, I don't know, but anyway, there aren't any.
It's a very exciting prospect. I'd like to finish on a sort of, on the important question of whether we really want to live to 200.
I don't know.
And in the sense of, I mean, you know, there's aging, but there's also the diseases of aging. So can you just talk a little bit about whether what you're doing suggests that we can live longer and healthier, or whether what you're talking about is a long, protracted old age that perhaps not many of us would want?
Yes, that's a really good question. One simple answer to your question is that in these long-lived mutants, the diseases of aging, pretty much all of them, are postponed, and when they happen, they're not as bad. And that would make sense intuitively if there's something about being old that makes you susceptible. So in the mutants, if you're not old yet, then you won't get the diseases. So that's nice. That's a good sign. And as I said in my talk, it also makes it possible to get a drug approved for a disease rather than for slowing down aging, which would be more difficult.
Which might be a hard problem.
Exactly. But still, the question of happiness and, you know, exercise, and if you can do things, and if you're cognitively there.
You've got your grandchildren, you say, but your great-grandchildren or your great-great-great-grandchildren, are you still enjoying them by that
Yes,
stage?
that's exactly right. And those are very subtle things. I mean, you can just look completely normal but be depressed. And even that would be terrible. But having any kind of ailment, so the bar is pretty high. And so actually what people like myself and people who are thinking about translating this to humans, we're not really focused on extending lifespan, not really at all, but just really on staying more youthful. And I mean, it would seem like really great if you could be one of these centenarians who looks really young when they're old and then just die in your sleep. That seems perfect.
It sounds great. I suppose it's too much to ask that you have one piece of advice for everyone who's watching this about how they should extend their own lifespan.
Actually, what occurred to me after I've been thinking about this for quite a long time is, you know, I don't think people do enough to make the most out of each day. I'm really serious. I think if you really try to make each day a wonderful thing, then you won't extend your lifespan, but you'll do what we're trying to do by making it longer. You know, you'll have a better life. And, you know, so that you can do right now.
Well, on that fantastic piece of advice, I think, thank you very much for doing this, and thank you very much for doing the Norman Heatley Lecture. Thank you, Cynthia.
Thank you very much.
telomeres are the ends of chromosomes, and chromosomes are the little tiny things in every cell in our body which carries the genetic information, and they're made of long DNA molecules. And every end of the DNA has to have a little protective cap on it, and that's what the telomere is. And it really does protect the chromosome, and therefore it protects the genetic material. If you don't have a proper protective cap, because the cap can wear down throughout life, and then suddenly it stops working, then the cells don't work properly and start sending the wrong signals to each other and to other cells around the body. We're very interested in how the wearing down of telomeres is related to people's health, partly because we want to know, is it just a biomarker? Is it just, an indicator and it's really other things going on? Or how much is it really causing declines in health, which then are seen as increases in, say, the chronic diseases of aging? But we have a pretty good idea now that it is causing at least some of the risks that people will have the chronic diseases of aging. The big three, the diabetes, cancers, cardiovascular, and of course, we think more and more about dementias as people get older and older. And the telomere shortness that you can see is predicting a higher chance of somebody dying in the future. It's not a tiny effect. Our cells have a certain amount of an enzyme, and we call it telomerase, and it builds back telomeres. Unfortunately, it's not super good at it in humans. It's okay. It gets most of us through, good long lives. But sometimes it doesn't work as well. And there can be many other processes that are wearing the telomeres down. Telomerase can build them back up with the help of lots of other factors in the cells. So what we'd like to know is how can we help that building up process? Well, we've been really interested in what roles nutrition and exercise and what we eat, what role does that have in maintaining telomeres? Because we do know that maintaining telomeres is playing a role in what our health is going to be. And that's the thing that really matters for a lot of people is what's their health going to be like. And there's a great name, it's called health span, which pretty much is the term that says, your health span is the time in your life that you stay pretty healthy and you don't have a real frailty or a disease or some real disability that, really affects your life. So we all want our health span to be as close as possible to our lifespan. So we're down to the basics. Exercise, good nutrition, trying to do stress reduction, recognizing we can't avoid all chronic severe stress. It's very exciting when you find people of like mind who have similar kinds of goals and ambitions, and when the two mesh, it's wonderful because different strengths are brought to the problem. We bring our strengths as our research expertise and knowing a lot about, over the years how telomeres work, and more through a lot of interactions with studies, how that's impacting on human health and wellness. And Shaklee has a goal of human health and well-being, and so that is a very resonant kind of role that Shaklee is playing as well. And so having that ability there so we can respond and make it happen right away, that's something that the Shaklee support is going to make possible. And that's huge for us. When you bring two very different kinds of strengths together, good things happen. And so I'm very excited that this is going to be something where, we really will make some true advances.
Regions referred to as telomeres. These telomeric regions consist of telomeric repeat sequences. The exact sequence of telomeric repeat can vary from species to species. The repeat sequence shown here is found on telomeres of human chromosomes. Since DNA replication can only proceed in the 5' to 3' direction and requires a primer, it is not possible for a DNA polymerase to replicate the 3' ends of linear chromosomes. If this problem were not corrected, chromosomes would shorten with each cycle of DNA replication. The addition of telomeric repeat sequences by an enzyme complex referred to as telomerase assures that this shortening does not occur. Telomerase is a unique and interesting enzyme because, in addition to having DNA polymerase activity, it also contains an RNA sequence that provides a template for the synthesis of telomeric repeat DNA. Part of the RNA sequence hybridizes with the single-stranded overhang on the DNA strand, leaving a single-stranded overhanging RNA sequence. The repeat unit shown here is from Tetrahymena, which is the model organism used in telomere and telomerase research. The DNA polymerase function of telomerase then synthesizes a DNA strand complementary to the RNA found in the telomerase and subsequently translocates to the end of the newly synthesized strand, and the process repeats itself. Many cycles of repeats can occur. Once telomerase has completed its function, DNA primase synthesizes an RNA primer near the 3' end, and DNA polymerase fills in the vacant region. A short region at the 3' end will remain single-stranded. The end result is that telomerase will have added many repeat sequences from a few dozen to a few hundred. This prevents chromosome shortening.
In 1991, 8 scientists entered Biosphere 2, an enclosed artificial ecological system near Tucson, Arizona. Their mission was to live for 2 years on food grown within the dump habitat to learn information for future Biosphere space colonies. 2 years later, nearly all of them lowered their blood pressure, blood sugar, cholesterol, and other health measures. Still, their skeletal appearance was shocking. The project, which Discovery magazine described as the most exciting scientific project to be undertaken in the US since President John F. Kennedy launched us toward the moon, was created with the goal of studying the interactions between life systems within an enclosed system. These interactions and life systems included the 8 participants and their interactions with each other. The underlying objective was to assess whether a similar enclosed living environment could one day be created on another planet to simulate Earth living conditions in otherwise inhospitable environments. Roy Walford was the only physician of 8 study subjects in the Biosphere 2. He is a professor of pathology at the University of California, Los Angeles School of Medicine. Walford was considered by many to be a trailblazer within the world of calorie restriction and aging research. He practiced what he preached, surviving on what is known as the Cron diet, which recommends 20% calorie restriction, or roughly 1,800 calories per day. This diet included beans, wheat, rice, bananas, and root vegetables like sweet potatoes and beets, all grown in an environment without toxic chemicals or pesticides. To study the physiological effects of life inside the biosphere, as a part of the study, The participants agreed to have blood drawn before entering the dome and multiple times while inside the dome. The participants will consume approximately 2,500 calories per day as part of their typical diet. However, due to a crop failure, participants were forced to eat only 1,700 calories in the first 6 months and were unable to increase their intake to 2,000 calories for much of the remaining time of the study. Months after the Biosphere 2 experiment ended, Welford published multiple reports describing the health changes he witnessed in himself and other individuals with him in a synthetic environment. As expected, all team members lost weight. Holding on to fat or even muscles was nearly impossible on their 1,700 calories per day diet, especially with the massive levels of daily activity that the crew members were performing as they tended the land. Their blood pressure dropped well below the normal values, 120/80, dipping into 89/58. Their ratio of total cholesterol to high-density lipoprotein remained the same, as both low-density lipoprotein and HDL decreased. HDL cholesterol almost halved, dropping from 62 to 38 mg per dl. Their skin began to turn orange because of the beta-carotene within their diet. Observers from the outside looked in to witness a scene that was eerily similar to the one described by the physicians in the Minnesota starvation experiment decades before. Faces were sunken in, leaving the appearance of discolored and tent-like skin resting squarely on bone, signaling the end of a strange series experiment and an unfortunate twist of fate. Welford passed away in 2004 at the age of 79, nearly a decade after exiting Biosphere 2. He was succumbed to Lou Gehrig disease, which he felt was precipitated by the elevated levels of carbon dioxide and nitrous oxide that he was exposed to during the time within Biosphere 2.
They're really beautiful creatures. They have really simple body plan and two googly eyes. They stare deep into your soul. they're great, cute little worms. They don't need a lot of work to be maintained. You just cut them, and every little fragment that you cut will make a new worm.
There is a whole host of biodiversity out there that we don't know about because we don't study the organisms that show that biodiversity. And so by studying planarians, we can find out about biology that we could potentially apply to humans one day.
In Peter Radine's lab at Whitehead Institute, flatworms, called planarians, allow researchers to explore the mysteries of regeneration.
We're very interested in what it takes to regenerate, sort of what are the minimal components you need to explain how an animal can regenerate something missing. Using this organism as a sort of experimental tool to probe the unknown, but we're also interested in generalizable principles.
Peter had been working on planaria for a long time, and he was interested in whether or not all these facets of regeneration that we see in planaria are weird quirks of planaria, or if they are actually necessary for regeneration of many organisms.
Radine's lab explores how a planarian creates a map of its anatomy, a system of molecular signposts that tells a cell where it is in the body. The genes that encode these signals are called position control genes. The researchers had previously found something intriguing. Muscle cells express the position control genes, and that gene expression then guides the regeneration of body parts in planarians. They wanted to know if other animals worked the same.
And the best way to get at this question is to go to something that regenerates but is really, 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 really different.
The researchers found a species that fit the bill, a marine worm called Hofstenia, also known as the three-banded panther worm.
Both worms, so you might think that they're very similar. Actually, they are incredibly different. We are more related to planaria than Hofstenia are. Hofstenia are as far away as you can get on the evolutionary tree before hitting jellyfish.
Planaria and Hofstenia had a common ancestor 550 million years ago. It's also the common ancestor of all animals with bodies that have two-sided symmetry, including humans. This shared ancestry far back in evolutionary history means that if Hofstenia also expressed position control genes in muscle, that could indicate that many other animals do so as well. The researchers observed gene activity in Hofstenia, looking to see which cell type expressed the genes for positional control.
And what was that cell type? Muscle.
In Hofstenia as well, muscle cells are also responsible for saying this is head and this is tail, and tell the stem cells then where they are so that they can make head tissue or tail tissue.
So that was very exciting to us because if, again, the similarity reflects common ancestry as an explanation for the similarity, then it points to a role for muscle in controlling regeneration and positional information in adult tissues dating back over 550 million years ago. If that's true, then it might be widespread in animals today.
But planarians have more than one type of muscle.
You have what we call longitudinal fibers that are basically muscle fibers that go from the head to the tail of the worm. And then you have circular fibers that they just go around the worm. And then you have diagonal fibers.
By feeding the worms RNA molecules that knocked out genes needed to make a particular muscle type, the researchers produced worms that lacked either longitudinal or circular muscle.
As a tool to then ask what are the roles of those muscle fibers in regeneration. We could inhibit the gene, the fibers slowly start going away, and then we could cut the animal and look at what happens.
So what we found actually that was very surprising, different subsets have different roles during regeneration.
Longitudinal muscles have a particularly crucial role.
Longitudinal fibers actually express a certain amount of genes once you cut the animal that are essential to regenerate, so they require these genes in order to regenerate.
So when we inhibited the ability of the longitudinal muscle fibers to be maintained, they slowly went away, we cut the animals completely failed to regenerate. So why don't they regenerate? Well, what we found was that after cutting them, the animals failed to reset positional information.
There's a position control gene called NODEM expressed in the longitudinal fibers that tells a cell whether it's in the head or the tail.
Based on the expression of this gene, the cells in the animals know what is the anterior, so where to regenerate a head versus where to regenerate a tail.
And this suggests that the resetting of positional information after injury to now re-specify the positional coordinates for the missing cell types is a driver of regeneration. So the fact that they couldn't reset positional information means they didn't regenerate because they didn't know anything was missing. Now, when we turn to the circular muscle fibers, again running from side to side, what happens when we cause those fibers to be lost from the animal?
Animals that don't have those fibers start duplicating the head. It's pretty amazing to see. We found that most likely it's because an anterior organizer that these animals have has been duplicated. We hypothesized that these circular fibers are required to constrict somehow that organizer.
So it indicated these different roles for different muscle fibers and controlling attributes of how regeneration occurs.
But ultimately, it's the stem cells that build new tissue, replace missing heads and tails, cell by cell. What rules did those stem cells use to decide where to form new structures?
So I'm very fascinated with the concept of self-organization, which is the emergence of structure and function out of local interactions between the elements of an initially disordered system.
The researchers asked if stem cells have self-assembling properties. They focused on how specialized stem cells called eye progenitors rebuild the eye, which was a small, discrete organ they could study in isolation.
If this is true for the eye progenitors, it's likely true for many progenitors for many tissue types.
The style of the experiments they used was a throwback to the classic planarian experiments from centuries past.
A lot of the experiments in this work could have been done with a razor blade and magnifying glass. It was really the concepts that were sophisticated.
The researchers wanted to figure out how stem cells respond to the positional information that muscle cells provide. When there's an injury, position control genes in muscle create a map that's a scaled-down version of the original body map. But in some uneven injuries, the recreated map is shifted relative to the remaining body parts. The researchers predicted that this mismatch could produce an animal with 3 eyes if the animal was decapitated, cut down the side, and one of the eyes was removed during head regeneration.
I remember the day we drew that shape on the board. We asked ourselves, can this really happen? And then we did this experiment nearly on the same day.
They cut the planarians down the side and decapitated them. While the head was growing back, they removed 1 eye and waited a few more days.
And then the moment we saw these 3-eyed animals was the most amazing moment.
These results showed the researchers that eye progenitors did self-assemble. They weren't solely relying on the body's positional map. They could form structures because they attract each other.
So self-organization allows the eye to basically serve as an attractor, stabilizing the function basically and the position of the eye, while the animal is restoring its correct proportions. So that's the first feature.
2 other features help explain why the stem cells formed 3 eyes instead of 2. The second feature is the target zone, created by the positional map.
Eye progenitors or any kind of progenitors basically read out this map to go and find where they should nucleate and start growing.
A third feature puts the first 2 together. It's called the targetable zone, which is the broad area in the body where organ progenitors hang out, ready to respond and rebuild structures in case of injury. It's a wide area, so that if an animal loses its whole head, it still has progenitors for organs in the head.
Imagine a small targetable zone right around the eye. Upon decapitation, we would lose all of it. When you have a broad target zone, this allows the existing anatomy to have access to these progenitors while the new tissues are being regenerated.
Even if that organ is out of place, with the coordinates guiding the placement of new tissues that were missing.
So the features of self-organization, the target zone, and the broad targetable zone let the researchers explain why they're able to produce these 3-eyed worms.
What we're able to find is a small set of principles that we think explain how tissue architecture is maintained and organized during the process of regeneration. A lot of our findings have necessarily focused on different pieces to the puzzle, different modules, but what's been exciting is to see how some of these modules fit together.
I particularly enjoy solving some small puzzles. I enjoy just the plain discovery of finding things and trying to understand how they're connected to each other, how they're working to each other. I think that's the fun of the basic science, trying to put the pieces together in a big, in a big puzzle.
The reason we can't fix a broken spinal cord today is we are missing certain fundamental answers regarding the biology. So that's why basic biology is incredibly important. So looking at these animals that are incredibly proficient in their regenerative capacity, I think we can uncover a lot of fundamental rules.
One of the reasons why we can't just, if you cut off your arm, it can't regenerate, is we can no longer set the positional information that says, I'm going to be a finger. And so if we could figure out what tissue could give this information, then maybe we could find a way to unlock that holy grail of regenerative medicine.
We have a sort of a draft explanatory model for how an animal could regenerate. Now, there's lots of holes in that model, lots of things we don't know, we don't have explanations for, but still, can start to see how it might work. And that is very exciting. And we've been pretty focused on trying to explain the logic of how regeneration works in planarians for many years, and it's satisfying to feel like you're starting to see how some key pieces to the puzzle work.